In a previous lecture, we discussed the process of mitosis, which is basically a replication of the cell's nucleus and most of the time the cell itself. Mitosis results in two identical nuclei and most of the time two identical cells. It's the way organisms grow, basically, and heal. This lecture is going to be about sexual reproduction. It's a little bit different, and it leads us into our discussion of genetics. Sexual reproduction requires two parents. Sexual organisms have two parents. Each parent gives its offspring one complete set of chromosomes. They have gametes, eggs and sperm. Each gamete, each egg, and each sperm have only one complete set of chromosomes. All the genes for the organism is in the egg or in the sperm. It's one complete set of genes on one complete set of chromosomes. A cell with one complete set of chromosomes, like an egg or a sperm, is said to be haploid. Haploid is abbreviated by a small n, and in some books it's called monoploid. Same thing. Since sperm and eggs do unite, they fuse together in the process called fertilization, or sometimes it's called syngamy. The fertilized egg is called a zygote. The zygote has two complete sets of chromosomes, one from each parent, one complete set from each parent. A cell that has two complete sets of chromosomes like that is said to be diploid, or 2N. It's good to have two sets of genes, because if one gene for a trait doesn't work properly, you have another one that just might. It gives you a nice backup set. So a lot of our genes may not work in our bodies properly, but since we have two sets of genes in each cell, one from the egg and one from the sperm, we might be able to back up the bad ones. Now, the two sets that you have are not identical. The two sets of chromosomes are not identical because they come from each from a different parent. And these are different people with different backgrounds and different genetics. If an organism's complete set is 3 chromosomes, like some flies have, by the way, humans have 23 chromosomes and a complete set, we say that the haploid number would be 3. 3 chromosomes is one set. And a diploid number, 2 sets of chromosomes, would be 6. Since chromosome number 1, let's say in the fly, from the male fly, is not the same as chromosome number 1 from the female parent because they're different flies, we say that they're homologous. They have the same traits on the chromosomes. The chromosomes are the same size, but the way the traits are set up might be different. One chromosome may have a gene for brown eyes on it, and in the same location on the other chromosome, there might be a gene for red eyes. All of our body cells come from the zygote, so they're diploid. We have diploid cells and a diploid situation in every one of our cells. The only ones that are not diploid in animals are eggs and sperm. Now, one of the questions we want to answer here in this lecture is, why can two parents have multiple children, but that are not identical if they all have the same two parents? Here's a picture of 2 girls that have the same 2 parents, but they're certainly not identical. So how is that? Well, the answer to that question has to do with a process called meiosis or meiosis. It's a little bit different from mitosis, and we're going to check it out now to see how that one works. Now, meiosis is a process. It's the one that changes those diploid cells in your testes and in the girl ovaries into haploid ones, into eggs and sperm. It's how you make the haploid one set of genes from 2 set of gene cells. You change diploid cells into haploid cells, and haploid cells only have 1 set of genes. So when they fuse together, it goes back to 2. But the interesting thing about eggs and sperm is that each set of chromosomes in them are genetically different. Each sperm is different from every other sperm. And if one sperm gets to an egg, the child will be born a certain way. If a different sperm from the same man and in the same sex act gets to the egg, the kid would be very different. And the same thing if it's a different egg. So every egg and every sperm is genetically different in people. A man might make 500, 600 million sperm in one day, every one of them will result in a child that's a little bit different from all the others. So how is that? How do you get that genetic difference? How is it that all these haploid cells that are made from diploid cells in the sex organs are genetically different? Well, the answer, of course, is meiosis, and we'll take a look at that. Now, this is the typical animal life cycle. We have a mature male, and when I say mature, I mean basically able to make sperm. It doesn't mean socially mature. In humans, it really is about beginning teenage or even 11, 12 years old. And you see it says 2N there. That means that all of the cells in that mature male are going to be diploid, two sets of genes. They came from the zygote or the fertilized egg that he grew from. Somewhere in his body, he's going to do meiosis. And in humans, that would be in the testes. When he does meiosis, he's going to take some cells that are diploid and change them into haploid cells, one set of genes called sperm. If you're a female, your body is also diploid. You have two sets of genes in every cell. She will do meiosis in her ovaries and create an egg, which is going to be haploid, one set of genes. If a sperm and an egg meet, if it's a human, it would be in the oviduct or fallopian tube of the girl, right after the egg comes out of the ovary. If the sperm and the egg meet, that process is called fertilization or syngamy. It's the fusion of two cells to become one cell. Each cell carries one set of genes. The sperm has one, the egg has one. And when they fuse together, you create a zygote. And the zygote obviously is going to have two sets of genes now, one set from the sperm from daddy and one set from the egg from mommy. That zygote is going to grow by mitosis and cytokinesis into mature males and females again, and the cycle repeats itself. Now, how does meiosis actually work? How do you create these eggs and sperm? How do you make these haploid cells from diploid ones? And how is it that they're all different? Well, it's not that different from mitosis. So let's take a look. An interphase, the cells are not any different from mitosis. It's just a regular cell doing everyday life. And if it's in the testes or ovary and it gets a signal to do meiosis, the first thing it's going to do is going to replicate all the chromosomes. It'll replicate the DNA. And DNA is replicated in interphase S, which is exactly the same thing that happens in mitosis, or before mitosis, I should say. And then in interphase G2, the two centrioles are getting to the ends, each end of the cell. They're going to start making the spindle, and all the enzymes and other things that DNA is going to work with are starting to get ready for the process. Then we get into prophase 1. Now, when I say prophase 1, that means meiosis has two sets of replications. There's a 1 and a 2. This one is prophase 1. It's the first prophase. And what happens here is that you have two sets of chromosomes. You have one set from mom, one set from dad. You have two chromosome 1s, two chromosome 2s, and two chromosome 3s. And in this diagram, you can see the black ones are coming from dad and the white ones are coming from mom. They are double chromosomes. They look like little H's because the DNA has replicated. Those are replicates. But the homologous chromosomes, the two chromosome 1s, the ones that are very similar, and the two chromosome 2s and the two chromosome 3s, they come together. They join. They call this process synapsis. And what's happening is that they're kind of sticking together. Chromosome 1 from dad is kind of sticking close to chromosome 1 from mom. Chromosome 2 from dad, the little dark one, is sticking close to the white one from mom, and so on. And same thing with chromosome 3. Also, it doesn't show it here, but the nuclear membrane is going to be breaking down, and those centrioles on the outside are going to start making the spindle for the chromosomes to move on. Now, one important thing you want to note here in chromosome 1 and prophase 1, besides synapsis, the coming together of homologous or similar chromosomes, is that when they're stuck together like that, they can actually exchange pieces. And if you look at that first chromosome on top, you see a little piece of white and black have exchanged places. That process is going to lead to diversity and one of the reasons why all the sperm are going to be different. And that is called crossing over or recombination. We're going to call it crossing over. Okay. Now, as I have it happening only in one set of homologous chromosomes up there, but it happens more than once, and even in one set, and probably in all the others too. And so there's many combinations that can happen here if pieces of chromosomes start changing places kind of randomly. Then we get to metaphase 1. When we get to metaphase 1, the tetrads, a tetrad is two double chromosomes, the chromosomes that have replicated, stuck to the homologous one. So it looks like 4 chromatids there. They stay together, and they land on the middle of the spindle, just like they did in mitosis, but this time as homologous chromosome sets. In other words, the chromosome 1 and chromosome 1 from dad, and the chromosome 1 from mom, chromosome 2 from dad, chromosome 2 from mom, and so on, still are together. And they line up together like that on the spindle metaphase 1. But because they do that, if you look at the other diagram on the right, you'll see that that's the same thing, except that middle chromosome there, that middle tetrad, that middle set of 2 homologous chromosomes stuck together, have tumbled onto that middle spindle fiber and landed on it the other way around. And if you can imagine, all the chromosomes can do that. They can just kind of like throwing dice. It depends on how they end up on the spindle. It's another way you can get all this diversity. And you'll see why when we get to the end of the process. This is called independent assortment. These chromosomes are lining up on that spindle independently of each other. And they can be on either side. The father one's on one side, the mother one's on the other side. And remember, we have 23 of these, not just 3 like this one has. And so you can imagine how many different combinations of the blacks and whites on either side of the spindle you can have. So independent assortment leads to a lot of diversity along with the crossing over. Then we get to anaphase 1, and anaphase 1 now it just all makes sense. The chromosomes finally separate. The homologous chromosomes or similar ones move apart from each other down the spindle to the poles or the ends of the spindle. That's anaphase 1. But there's still double chromosomes made of 2 chromatids stuck together because of the DNA replication. When you get to telophase 1, what's happened here is they get to the end of the spindle, and new nuclear membrane forms around them. They may or may not uncoil to become chromatin again, so you can't see it, and then they'll coil right back up again to do the second meiosis. But at this point, because you only have 1 chromosome 1, 1 chromosome 2, and 1 chromosome 3, even though they're double chromosomes, that's just because of DNA replication, but there's still only 1 of each chromosome. That's a haploid situation. We just made 2 haploid nuclei here. Then we get to the second meiosis, and the second meiosis is basically the same thing as mitosis. And what happens here, here's your telophase 1 right here. This is the telophase 1 cell right there. You see that. And what's happening here is that you get the metaphase, the nuclear membrane disappears, spindle forms, chromosomes line up in the middle of the spindle, 1 chromosome on each spindle fiber. They're double chromosomes because of DNA replication way back in interphase S. And then when you get to anaphase 1, they separate and they move down the spindle. And then when you get to telophase 1, they are moved to the ends, and they become the 2 nuclei here. And if you look at this nucleus, each one has 1 chromosome. So this is a haploid set, 1 chromosome 1, 1 chromosome 2, 1 chromosome 3. They're not double anymore. That's all because the double chromosome separated here. But you have 2. And if you look at it carefully, you see they're not exactly the same, because this one here has a little piece of mom's DNA here in this chromosome. This one has a piece of dad's DNA in this chromosome. All right. So when this thing becomes a sperm, these 2 are different. They're a little bit different. This kid will end up with a little more mom stuff, and this will end up with a little bit more dad stuff in his body and his development. If you go on this side, you see the second nucleus here is going to go through the same metaphase 2, anaphase 2. And by the time you get to telophase 2, again, you look at this one, these 4, if you look at the black and whites, these 4 different combinations of these guys' parents' genes are all different. This one, this one, this one, and this one are all genetically different. They all have one chromosome each, one chromosome 1, one chromosome 2, one chromosome 3, one set, nice haploid set, but they're different. This one has a piece of dad's. That one doesn't have a piece of dad's on chromosome 1. This one has a black chromosome 2. This one has a white chromosome 2 over here, you know. So what I'm saying is that you're making 4 cells here. They all have one set of chromosomes, but they're all genetically different because of the independent assortment of the chromosomes in metaphase 1 and the crossing over in prophase 1. So that's how that works. And depending on what sperm makes it to what egg, that's the kid you get. Okay. And remember, every sperm is going to be genetically different because of this, and every egg will too. So I hope that helped you to understand that. And we're going to go a little bit further than that when we talk about genetics in subsequent lectures. Have a good day.